Hello, everyone, and welcome to today's ASBMB Breakthroughs webinar, Quantitative Proteomics for Understanding Epigenetic Mechanisms. I'm Tyrone Lofton, ASBMB Strategic Growth Specialist, and I'm very pleased to be moderating this session today. This webinar is part of ASBMB's Breakthrough Webinar Series, which is sponsored by the ASBMB's Journal. Specifically, today's session is brought to you by Insights in Biochemistry and Molecular Biology, IBMB, ASBMB's newest journal. IBMB published notable advances in biochemistry and molecular biology. This includes biological chemistry, chemical biology, biophysics, cell biology, systems biology, multiomics, metabolism, and methods articles. Before we dive in, a quick reminder. You're welcome to submit questions at any time using the Q&A feature at the bottom of your screen. We will address them during the dedicated Q&A session after the presentation. This webinar has been recorded, and the recording will be available on the ASBMB website. It's now my pleasure to introduce today's speaker, Dr. Ben Garcia. Dr. Garcia is the Raymond H. Wittcoff Distinguished Professor and Head of the Department of Biochemistry and Molecular Biophysics at Washington University School of Medicine in St. Louis. His research focuses on developing innovative proteomic approaches to study protein modification and epigenetic mechanisms in human disease, with more than 475 publications. Dr. Garcia has received numerous honors, including the ASMS Medal and the HUPO Discovery in Proteomic Science Award, and is a fellow of the American Chemical Society, the Royal Society of Chemistry, and the American Association for the Advancement of Science. He also serves as editor-in-chief at ASBMB Journal, Insights in Biochemistry and Molecular Biology. Without further ado, please join me in welcoming Dr. Garcia. Ben, the floor is yours.
All right. Thank you so much to ASBMB for having me for this webinar. Thank you, Tyrone, for the introduction. And so it's a pleasure to be here and hopefully in the next 30 minutes, just kind of give people a little bit of update of what my lab has been up to over the last, couple of years. So some things that we've just published very recently and also at the end of last year. And I just want to also, as Tyrone said, kind of plug a little bit the new ASBMB journal, Insights in Biochemistry and Molecular Biology, IBMB. We are open and have our associate editors in place. We are putting together an editorial board, but we've started receiving journals, manuscripts, and so. I think we've just accepted the first manuscript ever, and we have several others, maybe about 7 to 10 that are in various stages of the review process. And so I would welcome everyone to think about, submitting your manuscripts to IBMB. IBMB hopes to be, kind of a companion partner of all the other ASBMB journals. And so if you have some manuscripts that maybe quite don't fit the scope of some of the other journals like MCP or JVC or the Journal of Lipids Research, think about IBMB. IBMB also wants to capture the discovery of your work. And so, if you have beginning stories that are emerging, some breakthroughs that you've made, methodologies, or in some initial insights into, potential new processes, we want to hear about those as well. So please submit to IBMB. And so, getting back to the science, and so today the title of my talk is Quantitative Proteomics for Understanding Epigenetic Mechanisms. And my lab has been very interested in applying quantitative mass spectrometry-based proteomics for understanding chromatin organization, chromatin biology, and also all the mechanisms that are termed epigenetic, which I'll talk about in a little bit. And so I think everybody knows that we have large amounts of DNA in most of our cells. And this is meter amounts of DNA that have to be compacted into a very, very small area of the nucleus. And the way that nature does this is to take this DNA and wind it on a ball of proteins called the histones. And so this is kind of, I guess, somewhat similar to taking string and winding it on a spool. And so this DNA, about 146 base pair, are wrapped around two copies each of these core histones, and this makes up the basic repeating unit of chromatin, the nucleosome. Nucleosomes are strung together by linker DNA and linker histone, histone H1. Nucleosomes that are found in more open regions of the genome and accessible are called euchromatic nucleosomes or regions. Those that are a little bit more compacted and more difficult to get access to, these are the heterochromatic regions of the genome. But this nucleosome fiber keeps folding and folding into our chromosomes, eventually into our chromosomes. For a long time, histone proteins were thought to be just structural static components of chromatin. But over decades now, researchers have found that histones are highly post-translationally modified by many different types of modifications. And so what you're seeing here is histone H3, just really the N-terminus. As you can see, a lot of different types of modifications, and many of these modifications occur on these tails that are thought to protrude from the surface of the nucleosome, because when the crystal structure came out, the tails cannot actually be crystallized. So they're flexible and thought to be kind of sticking out into space a little bit. And over time, researchers have found that these post-translational modifications are quite important. They actually regulate the histone's function with regards to gene expression and other processes. And now over time, by mass spectrometry and other approaches, there's been a whole host of modifications that have been found on the histones. You're seeing here probably just a snapshot, and this is probably out of date as well. A lot of modifications across the core histones. You have H3, H4, H2A, H2B, and H1. Many modifications that have been studied for a while, like methylation or acetylation, or phosphorylation of proteins. But you'll see a whole host of really interesting, exotic, and rare modifications as well. A lot of fatty acyl modifications, things like dopamine ubiquitination, simulation. There's all kinds of ADP ribosylation, all kinds of modifications that have been found on the histones, not just on the N-terminal tails, although I would say this is where most of the post-translational modification stoichiometry is found, but you can see there's modifications throughout the histone sequence. And additionally, you can see that several sites, there's hotspots, these lysines, and other residues can be highly modified, with even up to 10 plus different types of modifications. So this is a very complex kind of language of post-translational modifications as put on the histones, and one that has taken years for methodologies to be able to detect them all and to try to understand when these modifications are changing under different conditions, so that the biology can then be inferred. And I mentioned that these modifications are part of the mechanisms that are called epigenetic. So epi is Latin for on top of. So these are all the mechanisms on top of genetics that can affect gene expression. And so you have histone modifications and DNA methylation and also small non-coding RNAs as well. And histone modifications, again, you're in cartoon form. You're seeing some of the more abundant modifications, methylation, acetylation, phosphorylation on some of the hotspots of arginines and lysines and serines on the histones. And over time, researchers found that certain modifications were linked to specific gene expression states. For example, histone acetylation is linked to transcriptional activation, gene activation, whereas histone methylation, it's a little bit more complex. There's different degrees of methylation, and this could lead to both gene activating or silencing. And so if you look at histone H3 and this lysine methylation here, this is found on the promoter regions of active genes. So it's an active histone methylation mark. If you go a few amino acids down, same type of modification, methylation on this lysine right here on the same histone protein, this is actually found, this is lysine 9 methylation, and it's found on the heterochromatic regions or silence genes as well. And so you have methylation, two methylations on the same protein leading to two different functions just because they're found on different residues. And as this data started coming forward in terms of the function, a lot of theories and hypotheses have kind of been put forward, and one postulated now over 25 years ago by Brian Strahl and Dave Alice basically said that maybe these post-translational modifications on these histones, these methylations and acetylations, that are mostly on the N-terminus, but spread throughout, act as some type of binding platform that then recruit specific proteins to specific spots in the genome. And this is the first step leading to turning a gene on or off. And in support, several proteins that have specialized domains, chromodomains, bromodomains, other types of domains, have been found to bind specific histone modification sites on the different histone family members. And so it's kind of an exciting time because, every year more histone modifications and histone binding proteins are being found and their functions as well, linking them to both human physiology and disease are being revealed. And with the explosion of genomics data over the last, 15 years, several mutations to genes have been shown to affect epigenetic, players in human disease. And so, we see a lot of mutations to enzymes that modify the histones, to proteins that interact with the histones, or even the histones themselves get mutated in certain diseases. And all of a sudden now you should have a lysine, but you have a methionine, and you wipe out methylation all over the genome. And because of this connection, several companies have epigenetic oncology divisions. Eli Lilly, Merck, other big companies have had these or have these. And then there's several smaller companies that only focus in this area, in this space, such as Constellation or Epizyme. And many small molecule inhibitors are making their way through clinical trials and FDA approvals to treat, several different types of human diseases, especially cancers. So there's a lot of interest in trying to understand whether epigenetic mechanisms might be at play in your biological system. But I always tell people that you're working in a really great area when your field makes it into blockbuster movies. And so here's a film still of the Amazing Spider-Man movie, one of the older Spider-Man movies from a while back. So you'll see on the board all the science that you need to know to understand how to get superhuman, amazing Spider-Man powers. And so there might be some things you recognize here, equations and drawing schematics, but if you look really closely right here, you'll definitely recognize something that's quite important, and that's a nucleosome with histone H3 K27 methylation on it. And so whoever was in charge of putting the most important science on this board for this movie knew they had to include some epigenetic mechanisms there. And so my lab has for a while been really interested in development of mass spectrometry-based proteomic approaches for understanding histone modifications. And that's because the traditional approach is using antibodies in various different types of assays, such as Western blot or immuno assays or immunofluorescence. There's a lot of challenges with antibodies. If you have a good antibody, life is great. You can do a lot of really cool experiments, and we use them as well. But oftentimes you're trying to figure out what the antibody is actually recognizing. Sometimes it recognizes more than one protein, as kind of shown here. Here's histone antibodies against these different marks, against different histone H3 from different species and recombinant histone H3. You can see this antibody is pretty good because this mark doesn't really exist in yeast, and so it only recognizes the mark. This antibody is not so great. It's recognizing histone H4 and H3. And this antibody is probably not very good because it's recognizing recombinant histone H3. So it's recognizing unmodified histone H3. A bigger issue is what you don't see cross-reacting, because what you don't see, can really confound your ability to make interpretations of data. And so here's a peptide ELISA with synthetic histone peptides that all have serine 10 phosphorylation, and we're using a monoclonal antibody that recognizes serine 10 phosphorylation. There's two peptides that have acetylation and methylation right next to that phosphorylation. And you can see this antibody is really good at recognizing phosphorylation when it's by itself, but when you have these two modifications together, the antibody is compromised and can't recognize the modification very well. So because of this, we favor using mass spectrometry. It doesn't matter if there's multiple modifications. We're sequencing the peptides, such as we sequence analogous to DNA sequencing. We can do this very quantitatively as well. And so now for the existence of my lab, my individual lab, and even beyond and before that, I've been, very interested in just development, further refinement of proteomics technology to characterize protein modifications, especially histones. And so, how do we make these modification analyses more quantitative? We want to look at which combinations of modifications occur together because one modification by itself, it almost never occurs. We do a lot of understanding the dynamics of these histone modifications, the kinetics, how fast these modifications are getting put on and off, as well as combining genomics with proteomics to hopefully one day kind of pull out histone modifications from your favorite gene and read the modifications and how they're changing in human health and disease states. So I'll talk a little bit about some of the technology that we've been using. Let me get through some of this animations. Lately, we've switched to using faster scanning instruments, and one of them we have in the lab is the Xenotoph 7600 from SciEx. And when we first got this instrument, SciEx had published this technical note that they could quantify 1,000 protein groups per minute using microflow gradients, so higher flow. And their data kind of look like this. It's hard to see this, but this is 5 minutes right here. I had a postdoc, now my instrument manager, Emily, who replicated their data. So this is our data, looks very, very similar in a similar time frame. And basically, yeah, we can get about, 4,700 of 5,000 proteins in 5 minutes. And in fact, this is what we use as our QC on this instrument. We just inject, and if we don't get around 5,000 proteins, 4,500 to 5,000 proteins in 5 minutes, then maybe we need to clean the instrument or tune it or something like that. So with this, we were quite convinced we could create a high throughput platform for histone proteomics. Our gold standard was using nano liquid chromatography, a 55-minute gradient, maybe with a hour and a half total runtime. And this is what we used on high resolution instruments. But could we make a really fast, gradient, 5-10 minute gradient instead, increase the throughput? And this is kind of what the chromatograms of some of the baseline histone peptides look like. We're able to do this because, get through some of the animation. On this instrument, we're able to take variable windows. And so in some of the previous instruments we've had to use kind of fixed length windows and do data independent acquisition across the mass range. And we're kind of stuck doing this. But we know that there's certain bins of mass to charge ranges that have many peptides and some that have very little. And so we can kind of optimize this and be able to take smaller windows when we have a lot of peptides coming out and bigger windows when we don't have. And doing this allows us to, because this instrument scans so quickly, to be able to also use a very, very short microliter per minute gradient, maybe a 5 to 10 microliters per minute gradient instead of nano LC as well, and still keep up and get data. And so this is what some of the data looks like. Here's our gold standard 55-minute. Here's our 5 and 10-minute quick runs. As you can see here, in terms of peak shape, MS, you can see that maybe 5 minutes is a little bit too quick. This looks kind of like a little bit of a triangle, but 10-minute gradient looks pretty good, very similar to our longer gold standard gradient. This is important for us because oftentimes we'll have peptides that have multiple lysines and they can have an acetyl group here, 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 here. Oftentimes the different acetylated peptides co-elute, and we have to take a lot of MS over these peaks to be able to tell if we have one isomer or the other. So even with these fast gradients, we still have enough points across the peak that we can tell these two different gradients, two different isomers in this gradient. We can actually tell all the different isomers for this particular peptide. This is something we published in MCP last year. In terms of number of peptides and the CVs of those peptides, going from our gold standard longer gradient nano LC to our shorter gradients, we didn't really sacrifice a whole lot of peptide numbers and CVs. We're really quite happy that the data kind of holds up and we can go to shorter gradients. Maybe 10-minute is probably the one we prefer right now. Here's 100 repeat injections of the same sample. You can see at the microflow, 5 to 10 microliters per minute. Chromatography is incredibly stable. Has been reported by a lot of people. And so we have very good reproducibility, in terms of number of peptides, really very, very consistent across. And this allows us to quantify about 150 to 200 different histone modifications in less than 15-minute total runtime, which allows us to do a lot of studies that we couldn't do before, population studies and other things as well, really large number of samples. This instrument also has electron activated dissociation besides collisional dissociation on it. And this is important, of course, if you want to, interrogate large peptides or intact proteins, you could do that with this instrument. And we've done a lot of top down and middle down with this instrument. But for us, we also found another advantage of this in that there's a lot of parameters that you can tune with electrode activated dissociation that you can't do on other instruments. And you can go to really, really high energies, 20 to 30 electron volts. And we had a collaborator that came to us and said, we have evidence that we think there's an acetyl and a monomethyl group on a lysine residue on a histone. But we don't have the mass spec evidence for it, just a lot of antibody and other evidence. The problem here is that an acetyl and a methyl group is the same mass, they're isobaric to a propionyl group, a different fatty acyl modification. By chromatography, they elute out pretty close to one another, so it's really hard to tell the difference. When we fragmented them with EAD and looked at the fragments, they look pretty similar in terms of the overall fragmentation. But when we cranked up the energy, we were able to break off the side chains of these modifications. And when we looked at the low mass side chains that were breaking off, at that point, comparing to synthetic peptides, we can see that there's a difference between an acetyl methyl fragmentation versus a propionyl fragmentation. And this is the first evidence of two modifications on a single amino acid at the same time. That was published in Nature a couple years ago. I'm going to skip that and talk a little bit about a new project in the lab that we just got published in JBC a couple months or a month ago, a couple months ago. And this is looking at a new histone potential regulatory mechanism, histone proteolysis. So you can imagine if you have a lot of modifications and you wanted to remove all of them at the same time, the easiest way is to clip the histone tail and remove these all at the same time. And in fact, this would, really affect proteins that bind histone modifications to modify them or just to bind them or erase the modifications as well. And there have been a handful of processes that have seen clipped histones in the nucleus. And I had some talented group members that have been working on this. Previously, it's been shown that histone H3 is cleaved by an enzyme called cathepsin. And this also can drive senescence as well, this kind of cleavage. But, the actual mechanisms of how this is happening, why it's happening, how it affects gene expression is not very well known. And so when we started looking at some of our top-down data, just from histone H3 extracted from histones extracted from HeLa cells, we found that the proteolysis wasn't just histone H3 as been published before, but we saw a lot of cleavages this is actually a sequence of histone H2A. Some of these are really abundant. We thought maybe cathepsin is still the protease involved here. And so we took recombinant histone H2A, incubated it with cathepsin, and we saw a lot of the same cleavages that we saw in our vivo HeLa data. We saw recapitulated in vitro. And so from here, what we wanted to do is to take this embryonic stem cell system that a little bit of histone H3 cleavage had been found before, and look for this H2A cleavage. And so this is just differentiating cells into embryoid bodies and neuronal precursors with retinoic acid. Very, very simple to do. And when we do this and we blot for a histone antibody, we do see some cleavage sites on histone H2A coming up. They're a little bit harder to see with the antibody. But when we quantify these by mass spec, we see several cleavage sites such as L23, F25, G37. When we look at different days of differentiation from 0 to 2 to 4 days, going from embryonic stem cells to embryoid bodies, we see that there's some sites that increased quite a bit, either at day 2 or day 4, in terms of their abundance, these fragment cleavage sites. And so these are some of the ones that we mapped using kind of middle down, top down proteomics. So is cathepsin really the protease for this histone cleavage event? The protease itself increases in abundance during differentiation. So cathepsin comes as a protease, intermediate, and mature form. You can see the abundance increasing at day 4, which would be consistent with the cleavage that we saw increase at day 2 and day 4. If we knock down cathepsin, do we see a decrease of the cleavage sites? And in some of the cases, we do. So here's a scramble control and the knockdown. We can see at L23, we get a knockdown. G37, we get a knockdown. V27, we get a knockdown. Some of these other ones don't change, these other cleavage sites. It could be that there's other proteases that are responsible for these, or maybe there's, these are artifacts. But we do see with the knockdown of cathepsin that the fragmentation of many of these fragments abundance decreases. So, why would you want to, talked about this before a little bit. If you wanted to remove modifications, that's probably why you'd want to have a protease come in and cleave the tail. Does H2A have modifications on its tail? Yes, it does. It has acetylation at K5, K9, or K5, K9 together. Those are the most abundant ones. If we track these during differentiation, all of them actually go up at day 2 and then go down at day 4, up and then down, up and then down. And so what happens when we knock down cathepsin and then look at the acetylation abundances? And so these in blue are the data I just showed you, where at day 2, it's at a certain level and then it goes down at day 4, day 2, and then it goes down at day 4. Presumably, these modifications are going down at day 4 because the tail is getting clipped. But when we knock down cathepsin, the abundance of these modifications in every case doesn't go down anymore. That's because the protease is inhibited with the knockdown from cleaving these tails, so these modifications are preserved. If we do a genomics experiment called a ChIP-seq, where we use an antibody to H2A K9, and we pull out all the parts of the genome that have that modification. If we do this at day 2 and day 4, and then knock down cathepsin at day 2 and day 4, if you focus right here, we see that this peak right here that has the K9 acetylation goes down at day 4, just like it does in our proteomics data. But if we knock down cathepsin, that peak doesn't go down anymore. In fact, it actually goes up. And so you can see this here kind of on average, in the wild type, the peaks that have H2A go down, but if we knock down cathepsin, they actually hold or maybe slightly go up. What are all these peaks? These are all peaks that, these are all genes that are involved in differentiation. And so if histone acetylation is important and important enough to be regulated by protease activity, then there must be an important complex that recognizes that acetylation sites. At least that's our hypothesis. And so we did this kind of experiment where we took nuclear lysate, took synthetic peptides that had the acetylation sites on H2A or didn't have the acetylation site on H2A, and did a pull down to see what proteins bound to those synthetic peptides for both the K5 acetyl, the K9 acetyl, or both of them together. In every case, they were pulling down the same complex, kind of shown here. This is the switch SNF complex. As you can see here from Western blot, this complex tends to bind the acetylated H2A versus the unacetylated H2A. Switch SNF is really important, especially in the embryonic stem cell state, because this kind of keeps self-renewal. But once stem cells start differentiating, you don't need this switch SNF at those genes. And so if we look at the overlap of H2A K9 acetyl and two members of the switch SNF complex as well, we find that the members of the complex, ARID and PBRM1, tend to be found on genes, on more genes, together with K9 acetyl at the promoters or the transcriptional start sites than by themselves alone. And in those genes, those promoters, the expression is higher when you have ARID or this PBRM1 with the H2A acetylation than just the switch SNF members alone. And so we could do an experiment where we actually introduce the clipped H2A that has that cleavage site. And when we introduce the clipped H2A that has the cleavage site, and we do pull downs with different types of nucleosomes, we can see that PRBM pulls down the clipped, the full length H2A. So these are flag tagged nucleosomes. So if you do a pull down with PRBM, it pulls down the full length. But when we pull down clipped nucleosomes with PRBM, we don't see the flag signal. And so it can't pull down these clipped H2As because they don't have that acetyl site that PRBM likes to bind. If we now kind of knock down cathepsin and look at the levels of H2AK9, PBRM1, and ARID, we could see when we kind of, in the wild type, the levels of K9 kind of, increase a little bit. And here in PRBM as well, when you inhibit cathepsin, and so you have a certain basal level, you inhibit cathepsin, it goes up. PRBM, you have a certain basal level, you inhibit cathepsin, it goes up. ARID 2, you have a certain basal level of ARID on this gene, and you inhibit cathepsin, and it goes up. indicating that protease activity is regulating the binding of these switch stiff members to peaks that have H2A K9 acetyl. Last piece of data I'll show you. A lot of it's published if you want to read more. We know that clipped H2A nucleosomes have more acetylated sites on them. So if we pull down, flag pull down full-length H2A or clipped nucleosomes, do mass spectrometry, we see that the full-length nucleosomes have more silence marks, shown in blue, versus the active marks kind of in orange. This means that those nucleosomes must be turned over quickly. And if we do a pulse taste type of experiment going up to 8 hours, we can see that full-length H2A containing nucleosomes are quite stable, but these clipped H2A nucleosomes, you can see will get exchanged faster, and by 6 hours, 8 hours, they're completely out of chromatin, so they have limited half-life. And so through this, we've been understanding the role of clipped H2A in various processes and disease states as well. And just want to thank collaborators, people in the lab who did a lot of this work, as well as different collaborators at various institutions and funding as well. And I'd be happy to try to answer any questions if you have any. Thank you.
Thank you, Ben, for that insightful presentation. We now have about 10 minutes for questions. I'd like to remind everyone that you can submit your questions using the Q&A feature if you haven't done so already. I'll start off with a question,
Sure.
Ben. For H2A cleavage, what is the significance in the research when it comes to the neurological diseases?
Yeah, that's a really good question. There's a lot of human diseases that have either overexpression, mutation of cathepsin. So how that affects H2A cleavage, and gene expression in those diseases remains to be seen. And so, I think I could see a big link to this histone acetylation in properly differentiating cells during different neurodevelopmental pathways. And so I think, this cleavage may be important to that because I think, once the cues for the cells to differentiate to specific neurological pathways is given, I think this is one mechanism that strengthens that by kind of removing the switch-NIF complex from those nucleosomes that are probably driving genes that are more needed for stem cell renewal. And so if that mechanism is somehow dysregulated, it's possible, you might end up with some neurological kind of disorders. And so that really is remained to be seen. Yeah.
So you're asked, could you please elaborate on the selectivity of the capsaicins on cleaving different histones?
Yeah. So cathepsin has been shown to cleave histone H3. Um, from mouse embryonic stem cells. We use mouse embryonic stem cells as well. In our data, histone H2A was the histone that was cleaved the most. So the most abundant fragments we've seen. But we did see a little bit of some of the other histones being cleaved as well. Cathepsin itself is a protease that likes to bind and cleave kind of more hydrophobic patches. And so there are, some very short hydrophobic patches on on the other histones as well. So we do see cleavage sites at some of the other histones, but by far the most abundant fragments we saw, at least in this system, were on histone H2A.
Then this is an extension to that question. Have you looked at isolation of these histones under different physiological conditions?
Have we looked at isolation of the histones in their various physiological conditions? Is that
Isolation. I'm pronouncing it probably wrong.
the isolation? Is that what you're
Yeah.
saying? Yeah. Um, I mean, we periodically go back and kind of isolate histones from various sources and kind of just make sure that all of our protocols are still working well. When we started this approach, or we started getting this data, we were very skeptical because we did think maybe it's an artifact of the processing. right? Like, leaving histones in acid too long or something like that. Or maybe there's a protease that comes along somehow and is involved, is in the preps and is cleaving the histones, or maybe there's other chemical cleavages somehow. So originally, we kind of ignored this data for a while until, we started seeing a little bit of literature pointing towards the cleavages. Um, and so, it took us a while to convince ourselves, and especially I think once we looked at the differentiation data and saw that the levels of some of the fragments were increasing, that told us that, there's some regulation here. It's not just a chemical artifact or something of the isolation. But that was a good question that we really had to kind of thoroughly convince ourselves of. Yeah.
All right. Next question. Is it possible to target pull down a specific modification pres presenting histones? For example, if the IP histones with K9AC, can you profile co-occurring modifications via MS?
Oh, just in general? Yeah, definitely. So, I mean, us and other labs have done things like that where we did like a chip mass spec type of experiment. So, yeah, you can pull down, all the nucleosomes that have K9 and then use mass spectrometry to see all the co-occurring modifications with that K9 modification. You'll get a really good enrichment of the modification with the antibody if your antibody's good. It won't be perfect, but you'll get an enrichment. And then depending on the mass spec you use, you'll either get very direct. If you do middle down, top down, you'll get very direct data that says, okay, these are the other modifications present with K9. Or if you do kind of bottom up mass spectrometry cut into small pieces, then you'll at least get kind of an average of, oh, these other modifications are found on these nucleosomes that have K9. They may not be, unless your IP is really, really perfect, there'll always be a little bit of, well, is it possible that these are on, a little bit of artifact, or there are two histone copies per nucleosome. So it's possible that maybe the two histone copies don't look exactly the same, but you'll get a lot of insight from those type of experiments. And yeah, people have done a lot of those before in the past.
Another question. For those of us who are new to mass spec, can you tell us about the types of fragmentations, approaches, slash considerations like CID, ECD, etc., specifically for analyzing PTMs?
Yeah, so no problem. If you're kind of doing traditional bottom up proteomics where you're digesting to small peptides, either with trypsin or something else, CID is usually sufficient. With histones, it's a little bit trickier because histones are so basic. They have a lot of arginines and lysines. And so just digesting them with trypsin, you'll get too many cleavages and really small peptides that many of them won't stick on columns very well. You also get many peptides that may contain the same modification site because they'll just be different linked peptides. And then quantification becomes difficult. So what we've done in the lab, and I kind of glossed over it in the first part of the presentation, is that we've developed an approach where we chemically derivatize the lysine residues that are not modified. And that way, when we digest with trypsin, we only get cleavage at arginine. And that makes very uniform tryptic-like peptides from the histones. If you're interested in longer pieces, things that are longer than tryptic peptides, longer than 20 amino acids or so, you probably have to start using electron based methods, whether it's like ETD or ECD or EAD. They're all kind of similar, but those mechanisms with those fragmentation types allow you to break bigger pieces and get very even fragmentation. Whereas if you try to use CID on bigger pieces, you're not going to get the greatest fragmentation. You're not going to get the best sequence coverage. And so I think the goals just kind of depend. Do you want to look at combinations of modifications, either on kind of big peptides that are like a middle down experiment or on Taq protein? That's a top down experiment. You're probably going to be using either, electron based methods or maybe like, laser based methods, UV, PD, things like that. If you're doing small peptide work, CID is usually good enough, or HCD is good enough. Collisional fragmentation is good enough.
There was a question that you answered earlier. One of the guests said the actual word was lactylation. So the question was, have you looked at the lactylation of the histones under different physiological conditions?
We have on some, on like a very specific project where our collaborators knew the lactol levels were higher. In general, a lot of those, fatty acyl modifications or other types of really interesting modifications like lactylation, they're pretty low level. And so if you just break open cells, you may not see a lot of those type of modifications, those propionals or other things or lactal, unless the conditions are really favorable to produce a lot of lactal, lactate, some of this might happen non-enzymatically as well. And so, oftentimes, if you really want to study those modifications, those fatty acyl modifications, those other really unique modifications, you will probably have to enrich for those modified peptides with a pan specific antibody, a pan lactal antibody. I think that's how a lot of people kind of do it. Um, and so, yeah, you can definitely study them. We've looked at a few of them here and there. Sometimes we've looked at some butyrylations and some other things as well. You could see some of the very abundant modifications without enriching, but if you really want a good analysis, you're going to have to enrich with some specific antibodies.
Thanks to everyone for submitting such thoughtful questions. And Ben, thank you for a detailed response. Ben, I'd like to hand it back to you for final thoughts and key takeaways before we conclude today's session.
Yeah, no, thanks everybody for really good questions. I'm not sure if we got to all the questions. If you want, you can definitely email me if you still have questions. Um, Happy to try to answer. In general, especially if you're wanting to start using mass spectrometry on histone proteins. They're a little tricky, and it's taken us a long time to kind of work out a lot of the methodologies and some of the software, but we're happy to kind of point you in the right directions. And again, I'll just plug, looking for a wide variety of manuscripts, for our new journal, ASBMB's new journal, IBMB. And so please kind of consider us. I think for this first year, the publishing charges are going to be waived. So that's like a bonus as well. And with that, just thank everybody again for having me for this webinar.
Thank you so much, Ben, for your time and expertise today, and to all of you for joining us. Be sure to keep an eye out for the next month webinar, ASBMB's